Hello, this is Alex Rodriguez, your Lifeline leader. Today we will be going over the genetic code. This is the last week of Lifeline. First, we're gonna go over a couple of reminders, the last reminders, really. Starting with Symbio for genetics. This assignment is due on April 18th by 11:59 p.m. Then we have the spot surveys, which are already available, and you will be able to receive credit for them until April 21st at 11:59 p.m. You just have to take a screenshot of the completion and then submit it to Canvas to get this point. If you're registered for the in-person class, which yes, it's no longer in person, but if you're registered for BSc 1010 043, you are required to also do the survey for that section, and that way you can get your point. If you're only registered for lecture, then you can only do the spot for lecture, submit um, that spot evaluation, and then you can get 1 point. The week 14 PPP quiz is also due on April 21st at 11:59. Please notice that all these assignments are not due on a Saturday at 11:59 as it usually has been. You in fact have a little bit less time to do this assignment. So just keep that in mind because you don't have the usual 10 days that you get. And lastly, we also have the Unit 4 extra credit quiz that will be due at the same time on April 21st by 11:59 p.m. What is DNA? DNA is a carrier of information. It is made of two antiparallel and complementary strands that are paired together, and the two strands connect to form a double helix. What does antiparallel mean? In order for something to be parallel or antiparallel, both of those things need to have a directionality. The directionality that we refer to is this right here, where we have a 5' end and a 3' end. Now, this will make a little bit more sense once we get to a portion where we can explain the counting of carbons. But right here, you notice how we go from carbon 1, 2, 3, 4, 5. You notice that this loose end over here has a 5' end. It is the fifth carbon is on the loose end. While down here at the bottom, it would be the third carbon that is on the end. Now these are antiparallel because one goes from 5' to 3', while the other one goes in the opposite direction, from 3' to 5', and this is why we call them antiparallel. Whereas if they were both going in the same direction, 5' to 3', then we would call them parallel. Each of these strands is composed of many individual nucleotide monomers, and this serves as a blueprint for protein synthesis. We will see that process in just one little bit. But first, let's understand what a nucleotide monomer is. A nucleotide is just one of these bases. These are nitrogenous bases. We have A, T, C, and G. When we pair this with another one, that is what we call a nucleotide monomer. This is the nucleotide structure. Notice that it is not just the nitrogenous base. A nucleotide also includes the sugar that is attached to that. So, it includes a nitrogenous base, which is a purine or a pyrimidine, and a pentose sugar, that is the deoxyribose, as well as the phosphate group. Those three together make a nucleotide. And then a nucleotide monomer is two of these together, where it is the nitrogenous base that binds to another nitrogenous base. Nucleotide, nitrogenous base, pentose sugar, and phosphate group. These three combined is what make a nucleotide. In DNA, there are four important nitrogenous bases. We have adenine, guanine, cytosine, and thymine. Bases on each strand pair together via hydrogen bonds. Remember from back in Unit 1 when we were talking about hydrogen bonds. Hydrogen bonds are not real sharing of electron bonds like covalent bonds are. Hydrogen bonds are more about attractions. We have two purines. Purines are those nitrogenous bases that have two rings in their structure. And then pyrimidines, we also have two of them, and those are the ones that have one ring structure. There may be a third one of those that we might discuss a little bit when we talk about RNA. But for DNA, we're only gonna see two and two, two purines and two pyrimidines. Adenine and guanine are purines, while cytosine and thymine are pyrimidines. Here you have it. This is the way that they pair. Guanine pairs with cytosine, so it is a purine with a pyrimidine. And then the other pyrimidine pairs with the other purine. So T, thymine with A, adenine. G, guanine with C, cytosine. Notice that this pair of C and G makes three hydrogen bonds, while this pair with thymine and adenine makes only two hydrogen bonds. And adenine and thymine pair only with two hydrogen bonds. Let's continue talking about the structure. The pentose sugar. 5-carbon structure where the carbons are referred to as 1', 2', 3', 4', and 5'. This is a better image where we can actually count the carbons that make one of these sugars. So these are the two types. We have deoxyribose, which is the one that we see in DNA, and then we have ribose, which is the one that we see in RNA. That is, in fact, why we call it DNA. DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. So here we can count that the first carbon is right over here, second, third, fourth, and fifth. From this third to fifth carbon is where we get the directionality. So whichever end, if it is the 3' end that is loose, then that is what we refer to as the 3' end. And on the other side, we would expect to have a fifth carbon loose, which would be referred to as the 5' end. In DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. And I already gave you the names, which should help you remember which goes for which. Again, the D in DNA stands for deoxy, and the R in RNA stands for ribose. Phosphate group. This phosphate group is bound to the fifth carbon on the sugar. It forms a sugar phosphate backbone, and these are covalent bonds that connect phosphate to sugar and forms long chains of nucleotides, extremely long chains of nucleotides. DNA has millions of base pairs, and all of these nucleotides are connected to each other through these phosphate sugar bonds. Notice that the 3' and 5' ends are right here. We have a 3' end here, we have a 5' end here. Of course, once this bond is created between these two, then there are no longer ends, and we would only be left with the 3' end down here. That is until another nucleotide comes along and it binds to it. So nucleotides will always be added to the 3' end. So to the 3' hydroxy is what we call it. And because that is the case, DNA is synthesized or made from the 5' to the 3' direction. That means that nucleotides are added from this side to this side. So nucleotides are always being added here at this 3' hydroxy. And that is why we say that DNA is synthesized from 5' to 3'. Another nucleotide comes in, it leaves a 3' end. Another one comes in, it leaves that 3' end. Over and over again. So more on the DNA structure, or an extension of what we've already seen. We know about the 4 nitrogenous bases, and we know that they are connected to a pentose sugar as well as a phosphate, and that unit of a phosphate, a pentose sugar, and a nitrogenous base is called a nucleotide. Now, several of these nucleotides are connected to other nucleotides down here via phosphate sugar bonds. These are covalent bonds. That is what makes the backbone of DNA. But on the other hand, we have the nitrogenous base, or the whole nucleotide really, connecting to another nucleotide on the other side via hydrogen bonds between the nitrogenous bases. And that is what makes the monomers, the nucleotide monomers. If we talk about the directionality, we see that the structure of DNA is antiparallel, meaning that one strand goes from 5' to 3', and the other one goes from 3' to 5'. They go in opposite directions. And ultimately, all the interactions that are happening between the two strands are what end up making the DNA a double helix. Chargaff's rule. This rule states that DNA from any cell should have a 1-to-1 ratio of pyrimidine and purine bases. What does this really mean? The amount of adenine has to equal the amount of thymine, because adenine pairs with thymine, while the amount of cytosine must equal the amount of guanine. In humans, the 4 bases are present as follows. We have a 30.9%, or we can really approximate this to 30% adenine, while we have a very similar number of thymine, and this follows because adenine pairs with thymine, correct? So this 30 and this 30 gives us already a 60% as a whole. Now we only have 40% left. That should already hint that in us humans we're only going to have 20% guanine and 20% cytosine. Of course, the numbers are not that precise, but you can see how they are close enough together that it supports the notion that G pairs with C and A pairs with T. So the amount of A plus G, the amount of adenine plus guanine, has to equal the amount of thymine plus cytosine. Let's see if that checks out. We have 30, we have 20, that is 50. We have 30, we have 20, that is 50. It follows Chargaff's rule. So knowing that Chargaff's rule says that A has to equal T and C has to equal G. If you have 40% adenine in your DNA, how much thymine, guanine, and cytosine is present? You will see that if we follow Chargaff's rule, we can find the number of all the rest simply by knowing 1%. So in this case, 40% for adenine. We know that adenine pairs with thymine, and we know that according to Chargaff's rule, A is going to equal T. Therefore, we're going to say that thymine is also 40%. Now 40% plus 40% leaves us with 80% to work with. And we need the number of C to equal G. Therefore, we are going to say that C is 10% and G is 10%. There you go. And we were able to get all those numbers only by knowing that adenine is 40%. It doesn't need to be adenine that's provided. Any one of these numbers that is provided gives you enough information to gather the percentage of all the other three if you follow Chargaff's rule. DNA replication. Why do cells need to replicate their DNA? This is so that cell division can occur correctly. This is important for both mitosis and meiosis. This is what allows organisms to grow tissues and sexually reproduce. That is why DNA needs to be replicated. That is the basis of why one cell can turn into two, and then two can turn into four, and four into 8, and 32. And if you go back to when we were talking about human development, you will remember the 4 cell stage, the 8 cell stage, the 16 cell stage, the 32 cell stage, and so on and so forth. From that DNA replication that started with a single cell, huge organisms arise. Organisms that are comprised of trillions of cells. And that is because DNA replication is accurate enough that it allows for this to occur. Now, we have to recognize something that we like to call the central dogma. The central dogma in biology is is a sort of rule of thumb that says DNA gives rise to protein. DNA makes RNA, and then RNA makes proteins. That is the definition of the central dogma. From DNA, we get RNA, and then it is RNA itself that gives the instructions to make a protein. So overall, DNA is what makes protein if we save that step in the middle of making RNA. That is the central dogma. So DNA replication happens in a semi-conservative mechanism. And what does that mean? Let's see. Semi-conservative replication produces two DNA molecules. So let's just jump straight into what semi-conservative means. Semi-conservative means that this is a process that is not completely conservative, right? That is very easily taken from the word. But when we're talking about DNA, what are we referring to? What it really refers to is that only one of the original strands is going to remain in the end. Let's have a look. We are always going to keep one original strand and synthesize a whole new one. That is what semi-conservative means. You keep an old one and you make a new one from that old one, or you use that old one in order to make a new one. We are going to go over the process of how this occurs. It is a fairly complicated process, but ultimately, that is what semi-conservative means. You begin with an original DNA molecule. That DNA molecule is basically split in two, and from the original two strands, two new strands arise. And then, when these two undergo replication, we are only left with that one original still that is going to give rise to a new strand. And the new strand that came from this one is going to give rise to another new strand. And that is the definition of semi-conservative when we're talking about DNA replication. So DNA strands act as a template to synthesize a complementary strand. Each new DNA molecule contains one original strand from the template and one new strand. Variations in replication. So replication can happen differently depending on what the shape of the DNA is. So let's have a look. Prokaryotes have a single origin of replication. Now, let's think about why that would be. First, let's notice the differences. Prokaryotes have a loop of DNA, a single loop, a single chromosome, nothing but one loop. It is circular, okay? While eukaryotes, well, we already have been discussing since we started talking about mitosis that eukaryotes have chromosomes, right? That means that we have more than one molecule of DNA. In fact, we humans have how many? We have 46, right? We have 23 pairs. So we have 46 DNA molecules. Eukaryotes have multiple origins of replication. And that is not just because we have more than one DNA molecule. That is because even in a single DNA molecule, we can have more than one. And the reason for that is that chromosomes, or the DNA molecules that we have in eukaryotes, are linear. They have a beginning and they have an end. They are not circular. And that's one of those reasons. Eukaryotes' DNA is longer and it takes more time to replicate. That is really another reason of why we need multiple uh, origins of replication. If we had only one per DNA molecule, you can imagine how long that would take. As a general trend, eukaryotes have a lot more DNA than prokaryotes do. Multiple origins help speed up this replication time. Now, the mechanism of DNA replication. The initial DNA molecule has two complementary strands of DNA. Again, complementary because they match with each other, right? We know that A pairs with T and we know that C pairs with G. They're complementary because if one strand has A, T, C, for example, the other one is going to have T, A, G. They complement each other. The first step of replication is to separate or unwind the complementary DNA strands like a zipper. Each conserved strand now serves as a template for the addition of nucleotides along the new complementary strand. Now, this is very hard to picture, but thankfully, in the slides ahead, I have several animations that are gonna make this easier to understand. But for now, imagine a molecule of DNA, which you already know to be a double helix. The first step is going to unwind both of those strands. Each conserved strand is now going to become a template for a strand that is going to be newly synthesized. So remember, the mechanism is semi-conservative, meaning that each of those strands will give rise to a new one. And in the end, we're going to have two copies of DNA. Each one of those two copies with an original strand and a new strand. Nucleotides are connected to form the sugar phosphate backbones of the new strand. And that is done by something that we call DNA polymerase. It will come up ahead as well. According to the semi-conservative model, each newly produced DNA molecule now consists of one conserved strand and one new strand of DNA. Now, this is how the mechanism works. As you can see, it is fairly complicated. But the thing that I want you to take away from this image is not all the machinery that you're looking at here, because it is machinery, right? We have an enzyme here called helicase, which is the one responsible for unwinding the DNA strands. And then we have several others like DNA polymerase, DNA ligase, and so on and so forth. But what I really want you to take away from this image is that we have a leading strand and we have a lagging strand. And hopefully, as we look through the animations, you will understand why it is that we have a lagging strand and a leading strand. But we have already discussed the fundamental reason of why this happens. We have already established from the previous slides that DNA is synthesized from the 5' to the 3' direction. Alright? And that is why we have a lagging strand. Let's see if we can make sense of it looking at this image. So, remember, DNA replication is a semi-conservative mechanism. So we are unwinding the DNA here, and we have two original strands, which are anti-parallel to each other. Do not forget that. That means that one runs from 5' to 3', and the other one runs from 3' to 5'. Alright? So, knowing that DNA is synthesized from the 5' to the 3' direction, you can notice that this leading strand that goes from 3' to 5' is using this DNA polymerase to read the template strand and at the same time lay the nucleotides that match that leading strand. And those are being laid in that direction, 5' to 3'. That is what this strand would be. This becomes already a new molecule of DNA. Now, if we look at the other strand that goes in the opposite direction, from 5' to 3', we see that DNA polymerase runs into a problem because we already established that DNA is synthesized only from the 5' to the 3' direction, and therefore DNA polymerase cannot just bind here and begin producing new DNA in the same direction that the other one is, because if it were to do that, it would have to do it from the 3' to the 5' direction, and that is not going to happen. Alright, we already established over and over again that DNA is synthesized from the 5' to the 3' direction, and therefore in order to solve that problem where we do not have a polymerase capable of synthesizing DNA from the 3' to the 5' direction, what ends up happening is that this process comes along and fixes the problem. And that is a process where the DNA polymerase keeps synthesizing DNA from the 5' to the 3' direction. So it goes from right to left in this picture. It goes from right to left. But notice how this DNA polymerase is reaching a top here. And once it reaches the top, it needs to get out of the way, just so that then it can come back here and begin synthesizing all over again and fill in this gap. Let's look at the animations. So the first enzyme that we are going to discuss is going to be helicase. This is the enzyme that unwinds the parental double helix at the Y-shaped replication fork near the origin of replication. It breaks the hydrogen bonds between the bases. Remember that these two strands are being held together only by hydrogen bonds between the nitrogenous bases, right? The hydrogen bonds between C and G and the hydrogen bonds between T and A. Now, helicase comes along and it breaks those hydrogen bonds, and in doing so, the DNA separates. If you think of DNA as a zipper, then helicase is what unzips the zipper. Helicase uses the assistance of single-strand binding proteins in order to keep the strands separate. They stabilize the unwound parental DNA and hold the two strands apart, forming the replication bubble. Now, these need to be held apart, because if there isn't anything holding them apart, once they come close enough, they are going to reform the hydrogen bonds that helicase broke. And that is where the SSBs come in, the single-strand binding proteins. They allow enough time for the strands to be separated, and in doing so, they allow enough time for all the machinery to get through and synthesize new DNA. So here we have the first step. We have a double helix of DNA or a double stranded DNA, and helicase comes along breaking the hydrogen bonds between the nitrogenous bases and therefore separating both strands of the DNA. Now, from this point, we already know that one of these is going to become the leading strand, while the other one is going to become the lagging strand.